Spectroscopy is not the problem. Tuesday, April 14, 2026. Optica calls the Zoom room to order for 90 minutes. We're live that day. Time to see what survives the real world. Spectroscopy is not the problem. The problem is deployment. It shines in the lab, it sings in the demo, but can it hold when the pressure hits? Can it work in the medical surgery? Can it fit the pharma line? Can it read the field, the crop or disease, and do it right every time? Because performance is easy to praise when the setting is clean and controlled. But life is noisy, workflows are tight, and buyers want systems, not promises sold. So bring the diagnostics, bring biotechnology, bring miniaturization that actually scales, bring AI that clears the path, not adds complexity. This is where the real questions land. Reliability under strain, reproducibility across the process, usability again and again. Not just a signal, not just a graph, not just another clever result. Can it move fast enough, fit deep enough to matter where decisions are difficult? Spectroscopy is not the problem. Deployment is the fight. From brilliant measurement to trusted adoption, from prototype glow to operational light. So Tuesday, April 14, step into the Optica meeting, where life sciences meets commercial truth and good technology meets its reckoning. Because the winners will not be the ones with the prettiest data alone, but the ones who make spectroscopy useful in the places that matter most. is not the problem. Tuesday, April 14, 2026. Optica calls the Zoom room to order for 90 minutes. We're live that day. Time to see what survives the real world. Spectroscopy is not the problem. The problem is deployment. It shines in the lab, it sings in the demo. But can it hold when the pressure hits? Can it work in the medical surgery? Can it fit the pharma line? Can it read the field of crop or disease and do it right every time? Because performance is easy to praise when the setting is clean and controlled. But life is noisy, workflows are tight, and buyers want systems, not promises sold.
We're joined by an excellent group of speakers from these global companies. Reveal, Labby Inc., Agilent Technologies, Pioneers, Agate Sensors, Ipsen Photonics, Ligantech, Lyonics International, Nereos, and Clear Photon. Our thanks to Ligantech for sponsoring today's meeting, so that your online participation is free. So join our quest to build systems that people can trust, deploy, and use to make better decisions in the real world. Because that's where value will be created and faster adoption will happen. Thanks for joining us. So let's begin.
In the places that matter most.
Welcome everyone. Good morning, good afternoon, good evening, wherever you are all around the world. We're in a global meeting today to talk about a very important topic, to talk about spectroscopy and its applications on many different fields, from food and beverage all the way to life sciences. We need to find ways to increase the deployment of spectrometers. I would like to start by saying thank you to many of you. We are really seeing lots of people in the room already. More people are joining. But thank you especially to the company that made this meeting possible for everyone in the room today. Thank you all the way from Switzerland, the company Ligantech, offering silicon nitride photonic integrated circuit manufacturing and also heterogeneous integration with active components, including, of course, thin film lithium niobate modulators. Thank you very much, Ligantech, for bringing your silicon nitride platform to the online industry meeting today on spectroscopy. As usual, these are the rules of the game. We have two groups of companies. One group of companies who are the integrators. They're here with Santa Claus Christmas list, with a shopping list, looking for companies who can help them integrate the technologies that they need. And then we have the enablers, companies who are here providing a solution, and they are looking for an integrator to incorporate that solution for solving a bigger problem. Integrators today are the Chung Hospital all the way from Montreal, Labby, the company offering spectroscopy solutions for dairy products. Agilent, who doesn't know Agilent? The big Agilent is with us today, and coming all the way from Italy, Pioneers. And enablers, we have Agate Sensors, Ipsen Photonics, Ligantech, Lyonics, Nereos, and for the first time at an Optica online industry meeting, coming all the way from Poland, Clear Photon. We have actually organized these events for already 3 years, bringing every 2 weeks our corporate members together to do business. If you want to support what we do, and we try our best to do the best that we can, please join as a corporate member, optica.org/join corporate. Every 2 weeks we meet online. In 2 weeks, we are meeting online again to talk about a very important issue, thin film technologies for deployment in optics. Join our online industry meeting on thin film technology on the 28th of April, sponsored by OE Waves. And in the beginning of June, we all get together. We go to Brussels for the Global Advanced Manufacturing Alliance meeting with a spectacular agenda to talk about the future of lithography, optics in volume production, wafer level manufacturing, wafer level testing, wafer level packaging, the real solutions that our industry is demanding today. This meeting today is live stream in YouTube. So please look your best because you are on camera. Let me say hello to the YouTubers in the world. Hello, YouTubers. If you have any questions during the meeting, write them in the chat and I will read them in the room. And this, of course, also valid for the people here with me in the Zoom room. The next 90 minutes are for us to do business. If you have any question, comments, write them in the chat and we'll give you the voice for you to unmute your microphone and show how you can help, what you can do for them, and what they can do for you. And now let's start the meeting. I would like always to start the meeting with something very special. Dr. Helena Diaz has been working extremely hard to make sure that all of you on the next 90 minutes do great business. And she always focuses on bringing the right integrators, the right customers. And since this meeting is focusing on spectroscopy for life sciences, she chose a hospital in Montreal, Frédéric Leblond, representing the CHUM Hospital and Reveal Life Science. Frédéric, thank you very much for being with us. Thank you for being the opening speaker today.
All right. So thank you very much, Jose, and thank you, Elena, for organizing this and inviting me. So let me just see if I can. You see my full screen? I think you see my full screen now, right?
Yes, crystal clear.
Okay. That sounds great. So well, it's a pleasure to be here. So what I'd like to talk about today, essentially, is something that I think is very familiar to all speakers today, obviously, and it's really the clinical translational aspects of medical technologies here, with an emphasis on a technology that's called Raman spectroscopy, basically, and for surgery applications. And I'm representing, as Jose was saying, the CHUM Medical Center here in Montreal, as well as a company I co-founded called Reveal Life Science. So the problem that we're tackling with the technology I'm presenting today is really surgical guidance. So it's developing approaches using spectroscopy to use actually the large informational content associated with these optical spectroscopy technique. How can we use these information to help surgeons make decisions during the resection of tumors, for example? So there are multiple issues associated with that. One is just for multiple sites in the body where there are tumors, just the action of wanting to remove a tumor can be difficult, different depending on where it is, etc., etc. And also there is this aspect of tumors that is such that some tumors will invade beyond what we can actually see on I will say regular clinical imaging, basically. So if you see contrast on an MRI, for example, and you guide yourself as a surgeon to remove the total extent of that contrast that you're seeing on imaging, this may not be enough. And also, usually these images are required prior to surgery, right? So when you get into the OR and start digging and start removing tumors, for example, here, things will have moved, obviously. So the information that you got prior to the surgery might have degraded. So and here we're talking spectroscopy, and specifically optical spectroscopy. And it's and you will see there's a lot of speakers today that will be exploiting different aspects of what I'm mentioning here. But really, when you are shining light on tissue, what you're getting back can be interpreted in different ways and provide you lots of different types of information. So you can have fluorescence, either associated with the injection of fluorescent markers that will tag and target specific molecular processes, for example, associated with tumors. You can have endogenous fluorescence associated with like collagen, for example, that can trigger fluorescence associated with specific markers in the body. Also, like absorption, elastic scattering, as well as vibrational spectroscopy, Raman spectroscopy contrast that you can get. And the application I'm discussing is surgery. So you are in an OR, you want to use optical tools, but obviously there's light around, right? So this can be difficult. There's going to be interfering light, etc., etc. So this is an example of a constraint that's associated with bringing an optical technology in an OR for real. So that's just one out of multiple constraints that you will encounter, and that may come in the way of you being able to fully exploit all the information that the optical spectroscopy can provide. May it be like fluorescence that relates to metabolism, for example, of tumors. It could be like Raman information that's more specific and will provide you specific biochemical markers, as well as absorption and elastic scattering that will give you information about hemoglobin, for example, oxygenation of the tissue, as well as microstructures within the tissue. So ideally, you want to exploit all of that, but you want to be able to do it so that you transform this information into very simple outputs that a surgeon can use in real time during a surgical procedure. And that can be, as many of you know, I guess, that can be very difficult. So at Reveal, we've developed this for this application of surgical guidance, we've developed this console that combined with optical probes that were adapted to different surgical procedures. Like as examples here, we've developed this handheld tool that can be used during open surgery when you just point the device to a location, and within 1 or 2 seconds, you're getting this myriad of optical information as with spectroscopy that is transformed through machine learning into a single prediction. For example, saying like, is this tumor or not? This is oversimplistic, obviously, but typically that's what it does. So, and that tool was adapted for endonasal surgery, for laparoscopic surgery, for endoscopy also. There are different implementations that were developed. A strength of these applications, spectroscopy, and here I'm using Raman as an example, is not only its ability to potentially capture the biochemical information that could help distinguish between normal, so parenchyma, like normal tissue, and tumors, but it can also, and sometimes in a way that's easier than tumor versus normal tissue, it can help you detect the nature of different structures, right? So something that for Raman spectroscopy is trivial is bone versus muscle versus fat, for example. Okay, and you can generalize that across the body to detect these different structures and develop these approaches where you first detect, for example, where you are in the body, and then you can ask these tools to tell you whether that is tumor or not. The example I'm giving here is the tool that Reveal has been developing for neurosurgical guidance, specifically for the resection of brain tumors, including glioblastoma, these other types, these other gliomas, as well as metastases to the brain. And we've developed this workflow where that is totally seamless, i.e., there's a measurement that's made during the procedures. Now all lights can be turned on, within the OR. So you just make your measurement, 2 seconds, you're getting an output. And prior to getting an output, there's a stream of quality metrics that are evaluated to ensure that what you're looking at is the thing that the tool was designed to detect. In other words, if there's a little bit of blood covering the surface, as like extravasated blood, for example, it will pick it up. If there is contamination from other things, you're not in the brain. I mean, if it's a bad surgeon that's operating and he thinks he's in parenchyma, but he's on bone, it will tell you. That shouldn't happen, but it'll tell you anyway. So the workflow was developed so that we can provide this information in real time. This is an example here of this single point probe that is used to take multiple measurements during neurosurgery. And here it's a setup, right? It's just to show multiple measurements made one after the other in 2 seconds each. And here, this is an example of a real world use of the tool during a surgical procedure. This was for a meningioma done in 2025, at the end of 2025. And here I'm just showing the tool is not used yet, but the console, the console the computer that you've seen bottom left before was the display of the device. And now the surgeon will during the course of the procedure, now the surgeon is taking the probe, pointing to the area where they want the measurement, and the tool will output a prediction. In this case, it's cancer. it was confirmed to be cancer. And this can be done as many times as the surgeon wants during a procedure. And yes, so I'll just, I mean, this is a long video. I'll just go on. I don't think we're gonna learn anything new as we move forward. And that specific tool was tested, I would say, for real in a 14-patient study where the tool was used for surgical guidance. And we've demonstrated through that study that the negative predictive value of the tool was 94%. It was more than 200 measurements that were made, and the positive predictive value across glioblastoma, astrocytoma, metastases, and meningioma was in upper 90% plus. The PPV for glioblastoma was a bit lower, but there are reasons for that I can explain that are not related with the performance of the tool. So currently here in Montreal, the company, but with my lab essentially, are conducting 7 clinical studies simultaneously in Montreal. We're running more than 4 clinical cases per week with that tool here in Montreal. This includes the pediatric tumors, pediatric epilepsy, those children that need surgery for epilepsy. There's a subclass of that needs that basically. And we've generalized the approach also to endonasal surgery for a range of lesions in the paracellular area. And also, this is not something I'm talking about today, but we have also generalized to multiple, like for example, colorectal cancer, liver cancer, and other applications like that. The tool that we've been currently developing is one for breast cancer surgery, which is to assess margins during surgery. So I'm gonna go fast because I think, Elena, right? I'm gonna be running out of time fairly fast, right? So this is an example of
You're on time. Don't worry, Frederick.
Okay, this is an example of a device that was, I'm not showing everything here, but this is an example of a device that was designed to determine if on a specimen, here the example is breast conserving surgery specimens, if we can assess whether a given margin is positive or negative for cancer. And in the workflow of resecting a breast tumor, there is sufficient time to do that while surgeons are doing something else, so that we can, if we provide an assessment, and I know that there are other companies developing systems with the same intent, if there's a margin that's positive, for example, it can trigger the surgeon to remove more, send to pathology, and etc. So that's actually, there's a lot of potentials for these kinds of devices in our field. And this is just here a preliminary version of the device that we had made that just shows like the imaging of a full surface with Raman spectroscopy. And we've developed a machine learning model that can pick out basically cancer with more than 95% accuracy, sensitivity, and specificity. So that's it. So I just wanted to introduce these 2 products that we're currently developing to give examples of clinical need versus the requirements to translate these things and use them for real in the medical field. And also, this is not the topic of my talk today, but going from lab to real world use, and people here know about that, but this is very difficult. There are several things that we tend to do afterwards, after the fact, that should really be done before the facts. Like, for example, ensuring that there's a clear regulatory alignment with between what you're going to be developing and what you're going to be doing, ensuring that there's a network of KOLs, there's a network of people that can actually, your tool is actually going to be used, right? It can be the difference between something being used or not. I mean, it's very tricky, right? Sometimes you think you have a killer app, a slam dunk, but at the end of the day, for this technicality, it's not gonna work. There's a lot of and also how you interact with academia, how you make sure your IP is solid and is also something very important. And yeah, so that's it. I think I'm gonna stop here, and I'm happy to take any questions if there's time for that.
Thank you so much, Frederick. I mean, this is really nice. I'm very happy to see how Reveal is have been evolving since last 2 years since we talked last time, and there are several things here. So one thing you were talking about the adoption, but the other thing is this kind of techniques, and then as I put in my comments, the uncertainty is has direct consequences. In this case, that could be fatal consequences. So, what is the main source of variability in vivo?
In vivo variability,
Yeah.
it's not what you would think it could be. There will be lots of variability associated with these different optical techniques I'm mentioning, and they're not always the same. Initially, what we thought was that with these Raman techniques is that you would get lots of inner patient variability and potentially intra-patient variability with the signal, like the same type of tumor so, but what happens is that this is really low, the variability, basically. So we're getting a spectrum, and you have these anchor points in a spectrum that that have very low variance when you compare patients one with the other, and even within this a given tumor. And there are these other sort of let's call them optical markers within a spectrum that will have more variance. That variance could be physiological, but oftentimes it's related with the specific nature of the tumor mutational status and things like that. So you need to and sources of variability also are these confounds that could be like there's blood on the surface, there's a bit of light that contaminates the signal that you didn't think of removing, for example. There's things like that.
Yeah. So, and the other thing is like because it depends on what tissues you are attacking. So, uh, what resolution actually matters when we are talking clinically?
Facial resolution?
Yes.
Yeah, well, it depends, right? It needs to be, if you have a super highly resolved imaging technology that fits, let's just talk about this need for today, like surgical guidance.
Yeah.
And it works well within the context of what you're doing, right? It's great. The better the spatial resolution, in principle, the more information. But in this spectroscopy technique, there is always, I think, a trade-off between spectral and spatial resolution. And also, if your spatial resolution, like with these probes, for example, it's like 500 microns diameter. If it's twice as big, maybe you're gonna dilute the information you need, like the degrees of freedom you wanted to interrogate. Basically, maybe they're gonna be diluted amongst, So there's a trade-off to be reached, and so you see how I'm skating around the answer here, right? So there's no one answer, right? So if it works, it works for the application that you're developing.
Yeah, absolutely. So we have in the room Natalia from the Holt Center, TNO. So Natalia, you have a question for Frederick, and I know this is going to be a very good collaboration after this. So you guys have to talk a lot. Natalia.
I put it in the chat, Helena. So, Frederick, thank you so much. Very inspiring talk. Thank you. How do you see the technology uptake, scaling up this technology, and what are the barriers to have it in every hospital, every room?
Elena, the barrier is money. I mean, seriously, I mean, there are other barriers, right? But to run the trial you need for your regulatory submission, for example, that can be an important barrier, right? That can slow down the progress of a company, and even like make such that if you don't have any more cash, it makes you vulnerable to predators, right? So that needs to be done very, very carefully. But I mean, perhaps the most important part of this is making sure that you are ensuring that your product will fit into the right regulatory submission category basically? Is it a class 1, a class 2, a class 3? Is it you need to file a PMA, a de novo, 510K to FDA? So that can change everything, right? If you need to file a PMA, you'll be less attractive to investors than if you need your device only is 510K. It's not the end of the world. So yeah, so these are, I think, the most important barriers.
Yeah, so this is something that comes back to what you were saying at the end of your presentation, and is the adoption. So, what is the most limiting for the adoption?
Well, that I think everybody knows that here, right? But if you are providing you are making a product that will require completely changing practice, right? You're asking you tell a pathologist that you're gonna replace their what they're currently doing completely in a way. That's the I mean sort of with respect, I don't think it's gonna fly, right? So it needs to at least this is the way I'm looking at all these things that we're doing. We need to complement something. We need to provide added value to an existing process. Like if you can add a tool that's not too costly, basically that doesn't change the practice, really doesn't add time to a procedure, to a process, but it boosts specificity for detecting something by 15%, say, well, that can really fly, right? But if you're replacing it, I'm not saying it's not gonna, I'm saying it can be much harder, right, to replace. And also there's a lot of variability between centers within a country, between countries, between jurisdictions, basically in the way people are doing things. So if you want to fit everywhere, right, it can be very challenging.
Yeah, totally. So if we want to see beyond what is visible in the standard systems, something that we can have forward is using hyperspectral imaging. And I would like to invite Fabrizio Preda from Nireos, and he can talk about it. So Fabrizio, the floor and attention is yours.
Thank you, Helena. Hello, everyone. And thank you also, Frederic, for this very nice talk. So Frederic mainly concentrated on Raman and fluorescence. I will be mainly concentrating on hyperspectral, which I think would be a really complementary technique. So let me share the screen with you. We go in full screen mode. I think you can see the presentation now.
Yes.
Okay, so just to introduce what hyperspectral and spectral imaging is, basically spectral imaging is a spectroscopy technique which allows to capture an image of your sample over different bands. So these can be referred as RGB, where you have 3 bands, can be extended to the so-called multispectral imaging if you have 5 to 10 bands typically. And if you extend it even more, you can have the full spectrum of the light for each and every pixel of your image, and that is called hyperspectral imaging. And of course, this is a spectroscopy technique which allows, in some cases, to detect maybe some defects or some, say, portions of the sample that were not visible by eye. So you can really see some defects. Spectral imaging, of course, can be performed over many different spectral ranges, and in fact at Nireos we offer a portfolio of different hyperspectral cameras depending on the spectral range of interest, from the visible to the extended infrared. And in a specific example that I have here, which is pretty similar to what Frederic told you before, basically here we are using the Vis sphere hyperspectral system, which goes from 400 up to 1.7 micron wavelength, to image ex vivo tissues unstained. This is a breast cancer case. And you can see here in the image that the camera was applied close to the surgery room here to characterize the tissue that was extracted from the patient. So this is ex vivo and non in vivo. And by taking the advantage of the full spectrum here, I report you a couple of spectra curves of fatty tissue versus cancer tissue. And thanks to the fact that you are able to measure over a very broad spectral coverage here in reflectance, you're able to detect and differentiate, for example, lipids from blood, from fibroadenoma cancers and different species in the sample. And thanks to that, thanks to the different spectral absorptions of light here, we are able to produce such an image that you can see on the left here. That is the result of our hyperspectral analysis obtained in 20-second measurement time, where in false color you can highlight the tumor, which is in pretty good match with this histopathology result. Of course, this is a preliminary study that is not a medical device. So we have a study going on under this European project. It's called Spectroscopy, which aims at complementing the hyperspectral imaging, which is a fast technique, with the Raman technique, which is point-like but much more accurate. And the combination of these two techniques aim at developing and delivering a, let's say, complete system for this kind of margin characterization and tumor characterization during the surgery. As a very last point, I would like also to mention that this technique can be also extended to microscopy. So the hyperspectral camera can be also connected to any commercial microscope. And so you have the same thing. So your hyperspectral imaging, it can be done in reflectance, transmission, fluorescence, any kind of light that you have. And in this case, we are showing just an example of applying the hyperspectral image in microscopy on an unstained mouse lung tissue to detect and characterize the healthy tissue in the top from a fibrotic tissue here at the bottom. By coloring the image in false color, in green you can see the elastin, and in purple you can see the red blood cells, and we were able to detect them in autofluorescence thanks to the very tiny spectral difference in the fluorescence spectrum. So that's it. That's our 1 minute contribution. I would be happy to then take any questions from the audience.
Thank you so much. Thank you so much. So it's so let's see. Frederick, is this something that can be useful for you?
Yes, I'll email Fabrizio right after this meeting.
That's what I wanted to hear. And there is someone else here actually that is working in Raman. So Claudio from River D or River D.
Yeah.
Claudio, yeah. So how you can help here?
Yeah, I think we are also involved in the Spectroscopy project, so we will be providing the Raman module for that. So what River D already works with Raman spectroscopy for in vivo applications for more than 25 years now. We specialize in some applications, so for skin, but we have developed some prototypes for that are being used in hospitals for oral cavity cancer, and now with also for breast cancer with the Spectroscopy project. And what I would say is that all of the experience that we have collected over the last years put us in a good position to become a framework provider of Raman technology. So we cannot develop all the applications that we don't want. Also, we want to stick with what we are good at. So if you have a good idea for Raman application, if you want a modular solution that you can just plug and play, or if you have a project that requires a little bit more than plug and play, that's what we can provide. So reliable Raman modules and the expertise to translate that Raman spectra into actual information from concentration or further data analysis. So that's, I think, what RiverD can provide. So support the people with ideas and want to reach an application faster without having to dive into all the technical details and challenges that we had encountered over the last years.
Yeah, thank you. So Fabrizio, let me go back to you. So there is something about the hyperspectral limits, and what are the limits on the real-time hyperspectral?
Yeah, it depends, as usual, you know. So you can have different kinds of approaches to hyperspectral imaging. At the end of the day, with hyperspectral imaging, you are measuring a huge data. So you're measuring a three-dimensional data with a 2D sensor, basically. You have X and Y, which is the image, and the depth, which is the wavelength. and you're measuring that with a 2D sensor. So you have to pay somewhere. You can pay with measurement time, a long measurement time. This is on the order of 10 seconds, 20 seconds, and that's our limit because we do not have any other compromise in terms of spectral or spatial resolution. So these two factors can be as high as you want, and you don't have to choose a compromise between them. In other approaches, you can have a very fast measurement time, but you have to compromise between spatial and spectral resolution. So at the end of the day, you always have to find the best compromise. So there is no best, the best, spectral technology. You always have to find the best compromise depending on the use case that you are working with.
Yeah, makes sense. Makes sense. So this is let's talk a little bit. Then there is some challenges for the architecture itself. Okay, and what happened was we are moving on the sensors towards chip scale and software-defined spectroscopy. And someone that can help you with that is Michael Wedslund from Agate Sensor, all the way. Michael, the floor and attention is all yours.
No? Thank you. So yes, as a starting point, all current sensors are based on the simple principle of somehow separating light wavelengths from each other prior to detection. Either you do this through a push broom type of setup, as previously described, or then through this sort of filter-based approach. And this is sort of the way things are done currently, and specifically the push broom technology today is seen as the golden standard in this domain. Well, our approach to this is totally different. So our technology is relying on a semiconductor physics phenomenon that allows us to have a totally software controlled and defined capability. So our smallest sensor is a single pixel. And on our first chipset, we already have 256 software-defined spectrometers. And the size of the chip is roughly 7 by 7 millimeters. And this is not optimized in any way. So the smallest sensor suitable for a wearables type of device, for example, can be in the range of 2 to 3 millimeters in sort of footprint. And the capability as such is quite remarkable. So we are able to achieve same type of spectral accuracy as with a push broom. And considering the sort of capture time on this, we are able to do full rate hyperspectral video on this type of sensor and with the full accuracy. So this is a totally new approach, and we are really keen on starting working with companies deploying this type of new capability into all kinds of platforms and systems. So our approach to this is that we want to be a semiconductor chipset provider, and as this is totally software controlled and defined, of course, our offering includes both the hardware as well as the software in order to be able to integrate this then onto a device of some kind. And I'd be happy to take any further questions later on, but this was what I had planned to tell at this point.
Thank you, Michael. So that's one of the things. So is this like so limits of the CMOS on spectroscopy? So challenge. What are your major challenges?
So the sensor is bound to the band gap of the semiconductor material. So now we are commercializing on silicon first. So the bandwidth of the sensor will be roughly from 400 to 950 nanometers. Second generation of devices will then be ported onto 3.5 semiconductor materials, which will give us a bandwidth up to a bit in the range of 2 microns. So going into the sphere domain, but the first sensor will clearly be commercialized on silicon.
Okay, so I know Anton is around here. Anton, do you think you can help him?
Yeah, for sure. I think It's a very interesting technology, and I think for some applications we also can offer a platform that can be very very scalable. So we are most interested in scale. So yeah, please do reach out to us.
Very good.
Yeah. And we also have Xavier from Corning. Xavier, do you think you can help? Xavier? I know you are
Yeah,
around there.
yeah, let me unmute first. Sorry about that.
I saw you.
Yeah, thanks. Yeah, they're definitely interested to understand more also about the physics. I mean, I was kind of browsing through your website that as you were speaking, trying to understand, I mean, how it works. So interested to learn more. We can touch base later.
Yes, absolutely.
Okay, excellent. So yeah. So
Thank you, Elena.
no, sure. Absolutely, absolutely. So this is what happens. So we are moving now from all the way to the clinical. So we are going to go to very harsh environments with no control conditions somehow. And I would like to introduce you to Blavin Corporation, Julia Summerdin, who is measuring live what is happening with the milk that we are going to have. So Julia, the floor and attention is all yours. Julia, you are mute.
Here you go. So
Okay, here you are.
perfect.
Okay.
So good morning. Really nice to hear everyone's presentation. It was fascinating. I'm going to talk about something very different today. It's something dear to our heart in our everyday life, milk. I know some people don't drink milk, but you must eat butter, ice cream, and cheese. So that matters. So my name is Julia Summerdin. I'm the co-founder and CEO of Labby. Labby is a startup coming out from MIT. I founded the company back in 2017, 2018, after I finished my master work, finished my study at MIT. So over the next 10 minutes, I want to take you to my journey in the past 6-7 years I have been living to really figure out how to take the precision of optical spectroscopy and also image science, everything. We have a fusion sensor there, a sensor fusion there, out of the controlled laboratory and make work reliably every day in the working farm parlor, which is very different from our everyday environment. So that really turned out to be a much harder problem than it sounded, than I really initially understood. So that's exactly what I'm here for. I want to start talking about why dairy milk is such a compelling spectroscopy target. So you can see every milking, if I'm going to show you a photo there, you're going to see what the farm look like, but every milking van cows are milked 2 or 3 times per day. So carries rich diagnostic information. Milk composition responds dynamically to animal health, nutrition, stress, and also environment. For example, the fat content reflect metabolic balance. Protein levels track feed efficiency. Somatic cell count is biomarker for mastitis. Then we also have the milk speed that also encodes underlying genetic trait, which most people, even the scientists, didn't really know in prior years. So when you treat each milking as a data collection event, rather than just production event, you have this foundation for a continuous, non-invasive, and per animal health monitor system, and at a scale that does not exist anywhere else today. So I want to talk about a little bit about the problem. So the problem is very fundamental because for the past 50 years, the dairy still around all the testing diagnostic on the lab infrastructure pretty much from 20, 30 years ago. So you can see the process. Sample actually is collected manually by the lab technician who drive it to the farm, then ship it back to a central lab. So results return to farm. So that the testing only happen once a month. Then once that sample sent to lab, normally there are another 24 to 48 hours waiting before the customer can get a report. So when you think about that delay, number one, mastitis is a very common and expensive disease. Estimated about $32 billion loss due to mastitis, late detection and other production, drug treatment. But also antibiotics overuse. That's a bigger problem for not just for animal, also come to our food supply chain. So estimated about 70% cow treated blindly without understanding the cow's potential to self-cure or not. So another is we talk about today with dairy got a lot of attention because of sustainability. So 90% of dairy's emission are Scope 3. That means directly coming from parlor. How we treat the cow, how we feed the cow, eventually all become Scope 3 methane emissions to us. So industry today have no way to capture that as well, or even monitor. There's a lot of newer technology into new feed ingredients. So this is what the gap looks like. There's about $150 billion pound of milk produced annually in the US, right? The vast majority only merit that milk at the bulk tank. So once the milk go into bulk tank, that's 2 hours too late. So hours days after milking. And in the US alone, there are $2 billion loss per year to just subclinical mastitis, feed mismatch, and the quality downgrades. That was entirely could be detectable if you have right sensor, right technology in the right place. As I said, the lab spectroscopy community has solved analytical problem in the lab. The meat, marrow, butterfat, protein, SEC with excellent accuracy. That's not a question. But challenge the changes in environment today. We need to test milk, test cow in real time. You cannot wait for once a month, waiting for 8 hours for the results. So the lab instrument had to work in controlled, homogeneous, and thermostable conditions. A dairy parlor offer about 5. Dairy parlors is different. The degree varies from 5 to 35 degrees of Celsius, and also there vibration from pumps, there are biofouling, there are sample residue. So it's a very different environment than what lab original set out. So that's the problem we try to solve. On the high level, this is what the product look like. So we are the lab providing an inline optical based milk testing. So this is our inline optical sensor. It can directly, it's non-invasive. So we draw sub-sample milk from the milk pipeline into our milk stations. And from there, milk will be tested in real time under 10 seconds. So today we provide butterfat, protein, somatic cell count, also milk yield, and a flow curve. So it's completely non-invasive. The cow doesn't know it's happening. The farm does not change in a single step in their routine, which is really critical for product adoption. Because the sensor placed at every station, so you got individual animal data, not just diluted bulk tank average. This is an illustration what the deployment look like in the real parlor. I have a real photo later, you can see. So our sensor installed at each milking stations, cow side. It's real time, 3 times a day, twice a day. We also have a dashboard that updated results in real time, and you can see on the bottom. So farmer manager can check from the phone or tablet or any remote computer browsers. So looking at the session, you can see, this example, 4.2% butterfat detected, 3.4 average protein. That's on the, we can detect this to individual level, but we also provide bird eye view for the farm. So farmer knowing how their farm performance over as a whole. It's about 100 They produce about 1,204 pounds of milk. The SCC is about 100K, which is, healthy considered, could be better. So the resolution and the frequency, the herd visibility, without a device or technology like LabVIS, it does not exist if you use a lab monthly testing. So I want to walk you through the technical process. So during milking, LabVIS captured multi-channel optical data from raw milk stream. So that including spectroscopy information, also their microscope camera. So we look at colors, shapes, transition states, real time from a raw milk stream. And the edge model running could be entirely on the cloud or device, process signal in under about 10 seconds. I would say right now we can do under 10 seconds and deliver composition results. So this is, edge is important. So many farms today, they have internet, but not high-speed internet. So be able to process information, pre-processing data is critical, particularly when you have large data, camera data. So this is actually example of how our device work on the farm. You can see there are poos everywhere, there are peas everywhere. The device not only had to provide useful information, diagnostic information, but really had to be environmental proof. So that's what I'm going to talk about here. Those are challenge what the lab really doesn't teach you when I first get this one. There are so 4 challenges that really make, I would say, challenge the lab calibrated model when you bring to the lab. So first is optical fouling. The milk fat adheres to the optical window, within hours continuous exposures. So be able to use CIP cleaning in production. So really important because cleanability is the lifeline of optical sensing. When the optical window gets dirty, and ultimately you couldn't do anything. So another I was also add here is scattering. So milk is turned out really challenging for scattering. And how you remove that scattering, today we haven't found much way you can do mechanically. So you really had to use algorithm calibration to identify how to reduce or lower, eliminate the scattering effect on your model accuracy. So secondly, detect the drift. We don't make our chip. We use a third-party detector chip for our product. So that's also has a limitation. But overall, you're going to see LED drift. You're going to see sensor variation from side to side, from farm to farm, from time to time. So in order to overcome that, I would say what we learned on the field is there's no magic there. Large volume data really help us. So today we have tested about over, I say, thousands of cows, collect probably 50, 60,000 of data set to calibrate our device. So once device will be calibrated in the lab before we ship out to customer. So it does not provide the best performance because each farm is very different. So our goal is to provide robust, usable model. Once again on the farm, our model will be able to self-train based on new data come in. The model will be able to learn the new data stream and fine-tune, adjust the model based on each farm setting. So I would say third is talk about and as the milk is not the homogeneous materials, right? You have a fat globule diameter range from 0.1 to 15 micrometers. And also there are air bubbles and flow rate create signal variance that destroy reference standard calibration. That's why traditional lab equipment doesn't work there. So for us to overcome that is we do calibration, including repeatability calibration, and we also check the reproductivity, that just make sure device can provide reliable signal, because otherwise garbage in, garbage out before the data going into the machine learning model. So here I want to talk about one of the bigger cases for our technology is mastitis, because it's very expensive, it's very pervasive. So average farm have between 20% to 50%, depends on the region of the farm worldwide. So with monthly testing, as I said, subclinical mastitis, you miss early invisible symptoms that goes undetected for weeks. By the time clinical symptoms appear, milk already going to quality already be affected, and also contamination happen among the herd, right? So in many cases, antibiotics could be already given without giving the cow a chance to self-cure. So with LABI, the big difference is the farmer can see the somatic cell count at individual animal every single day. And the subclinical mastitis can be caught in the window, where the early intervention is the simple and is less expensive and also highly treatable. So that's a shift we see from really reactive lab testing to more preventative dairy management using LABI technology and the solutions. So I'm going to leave a question there. Here I want to say spectroscopy, maybe just say spectroscopy becomes operational tool when the system is designed for the environment. It's not as the other, it's not an environment for the system. So that's really the biggest lesson we learned. We had to really do all the adjustment to make our product work on the farm environment. Thank you.
Thank you so much, Julia. So actually, it raised a lot of interest here. So we have Mark Carmensin from Carmensin Solutions, which they specialize in contamination control. So Mark, could you please open your mic and say your question yourself? Mark, are you around still?
Yeah, it's just slow to respond.
Yeah, you have a couple of questions, so yes.
Yeah, so just one comment is, I just wonder if this would ever be useful for blood monitoring during blood donations. So that's just a future thing to think about, including things like anemia. And then how do you clean the sensor between cows in case one was infected?
Yeah, so that's a cleaning process. So the CIP, we do not, that's integration is really critical. You do not want to create extra friction to the working flow. So we basically, the cow, the farm clean 2 or 3 times a day. So at the end of each milking sessions, right, when they, like morning cycle, the whole system will be cleaned. That's independent of what we do. The farm does that for regulatory requirements. So the detergent in a very high temperature going to flow through our device, had to be flow at every corner, right? So that's how we utilize the existing CIP cleaning process. Otherwise, it's impossible. If you create an extra cleaning process, tubes, everything, not only you'll trigger the regulatory, red tape, but also that's just very expensive. So how to adapt your current product to work on the farm flow, that's really #1 lesson we learned in the technology adoption.
And if you have trouble cleaning, you might consider having an ultrasound cleaner to help aid the cleaning process for haze.
You can see once the device is embedded, all closed, right? So that's very different from lab, benchtop, from farm. Once on the farm, it's other hands. So unless you have, we thought about have additional cleaning capability to add on inside the device. But it's trade-off, right? That's going to add additional complexity to the device. The more electronic components, the more failure points you have. So the best way we learned is we have been tested on the farm with a commercial farm. There is some residue because the good thing with the pump, the force, right? So even you have a little residue in the beginning, but the cow got about 30 pounds every 5 minutes. So it's a lot of volume that flushes through our tubes, through our device. Most time that could remove completely the residue, in fact, or even any residue from within the optical windows.
Yeah. So we also have here in the room Henrik Madsen from SPIO Systems. So SPIO Systems, they create optical systems and micro optics. So Henrik, could you please open the mic, tell what do you do and what's your question? Welcome to the room.
Thank you, Helena. We are producing optical devices on wafer, meaning that we have a scalable process platform. My question is to you, Julia, is you are monitoring each cow on other level. Would it be beneficial to monitor each teat? So we have 4 individual monitoring for each cow?
Yeah, so if you recall our product, we do not touch cow directly, because that's why is the point, because once you touch cow, their animal welfare touch. So really what we touch is milking tubes, right? So if you do that per teat, which I know GIA, a German company, does that, it's become a lot more difficult, expensive. Number one, a lot more expensive. Number two, that create actual regulation points, right? But overall, yeah, I mean, for treatment, you want to know which teeth is affected. But overall, today we're not even there yet. First degree is you want to prevent the bad milk, subprime milk, going to the bulk tank, right? Because once you go into bulk tank, you have to say dilution is the solution. That's your, hoping, but that's too late. So for farmer, the first step is segregate the cow, separate the cow from the herd, divert the milk away from bulk tank, right? Then you think about treatment. And many times today, that's next stage we're also working on. You got to decide what the pathogen is contagious, non-contagious, right? Meaning the rule of thumb is if not contagious, 50% cow can self-cure. So you would want to give a chance. Then if a contagious, I mean, today with the regulation, you have to determine what is traits, what type of bacteria, the virus. So you want to do a pathogen detection, determine what's most useful by antibiotics to give into animal, right? So a lot of things happen there before you determine which teeth. So that's optimal, that's ideal situation, but that will add additional complexity. It's not the most needed in the industry today.
Yeah. So we have actually this kind of eclectic question. So Matt Singer from Thor Labs. So Matt, could you please open your mic and say your question yourself? Matt?
Yep, can you hear me now?
Yeah,
Thanks.
welcome.
I'm actually going to keep my camera off because I'm home with my infant today. But yeah, my question was related to infant formula safety. So there's been a lot of news recently of E. coli contaminations and other things with infant formula. And I wonder if this technology can be used in the home to detect, safety levels in the formula. This seems like you've solved a lot of really interesting challenges with getting these signals off of a highly scattering media such as milk. So I wonder if there's a new application for this existing technology.
We actually got approached by baby powder company 6 years ago. During the processing, they want to do live quality control, right? So I would say there are lots of potential there. I believe we can. The challenge for that, for each application, you got to collect enough examples to build a model, right? So I was saying for powder, it will be easier because you can just collect powder. You don't have to go on the farm, stand behind the pool of the cow. As a matter of fact, in the early days, the product can detect the blood in the milk. We also had other applications, nutrition, freshness of the fruits. So this is our product based on UV-Vis and image science. So the signal is detectable with the right excitation or fluorescence. So we're thinking about that, bring the product into the grocery store. But that will be need extra funding for us to dive into new application, because each application take years of domain knowledge. For industry to be, the domain knowledge is a critical important. You don't have a network, you don't have a partnership, you cannot land your product even on the farm. So yeah, we're interested. That's actually opened my ask. Is, as I said, we don't build our own chip, right? So we constantly evaluate third-party chips. So we're interested by chips that can provide a better spectral resolution, give us a broader range. So right now we're, in the UV-Vis, NIR, but if someone have a chip can from a width all the way to 50 nanometers, that will give our product a big boost, because that gave us extra signal there that currently is not available to our current sensors. So if you have a new sensors, chip-on sensor, biosensor, I'd love to talk to you guys.
So there is actually a lot of people in the room that can help you with that. But
Yeah.
before we go into this, I would like to call Anton back. So Anton Vasiliev from Tech. So you have your question, and actually this is a good moment for you to tell us what Tech can do in spectroscopy for life science.
Oh
So
yeah,
Anton,
there's an agent, that's a legend in the industry.
Yeah, hello. Thanks for introducing Helena. Yeah, maybe just I'm gonna go back to the question I said. So it's very interesting that you have this issue with scattering, and I was wondering, of course, you can also average your signal out for a longer period of time, but it would be interesting to understand, maybe you can also have more sensors in parallelized in one sensor that can measure the same. So you would increase the averaging effect. Maybe that would also help. And
Yeah,
yeah, go ahead.
Oh no, go ahead, finish. Sorry, I think that's a really interesting approach. We thought about it. Yeah.
Yeah. So let me share my screen. I can briefly say what we do. So we are a chip provider for with silicon nitride, and also for spectroscopy and life science applications. This is a kind of a snapshot of things that are mostly relevant for this crowd. So we have our base platform is based on silicon nitride, and we have a very low loss in different wavelength ranges. So we have a very good performance in the C-band, and also we have I would say very low loss in multimode waveguides as well. We also have results. We also have waveguides that work in the visible, so all the way down to 450 nanometer. And all the way up to 2.4 and beyond. So it's really limited by the platform itself. So it's silicon nitride and oxide. So in theory, we could go to 4 micron, but we have some extensive measurements done up to 2.4 micron, say. We have a modular platform, so we offer sensing modules, which I think will be quite interested for you. So where we selectively open the waveguide, so the bottom middle image, you see that there's an example where you can open up the oxide to expose the silicon nitride waveguide, and we have some very good performance there on that module with losses down below 10 dB per meter in that opened region. And so all these modules, we can combine them to offer more complex circuits or more complex functionality. So by integrating also photodiodes, modulators, or gain chips, you can think of having an integrated solution for an OCT or an ECDL or Raman source. This is an example here for an electrical nose that we're working together with Olfaction, who is a partner in Photonics Fab. And also, what's important for us is that we value our customers, and we have a story here together with Delta Life Science, a success story. They're quite happy with the path that we took together from prototype to scalable manufacturing. So we offer a very stable and reproducible platform. So you can invite you all to have a quick read on our website about the story, where Delta Life Science and a spectroscopy company or sensing company that from Netherlands that is working together with us to bring this product that they're making to volume.
Yeah, thank you so much, Anton, and that's the foundry and the scalable manufacturing platform, which is part of important because at the end this is part of the deployment. So now we need to go a little bit on what is going to represent this instrumentation layer. Okay, so this is where the performance must scale, standardize, and operate across multiple applications. And I would like to call to the floor Travis Bird, all the way from Australia. Agilent is present in the room. So Travis, the floor and the attention is all yours.
Excellent. Thank you very much, Helena and Jose and Optica, for allowing me to speak to you all this morning. I found the earlier talks absolutely fascinating and a great variety of uses of spectroscopy in life sciences. I would like to first just by giving a little bit of coverage of where the topics that I'm going to cover this morning or this afternoon for you lie. And I thought I'd first represent it by spectroscopy value chain in the sense that Agilent is involved in providing scientific instrumentation to the mining and the raw materials area. And what I'd like to talk to you about today is both the life science products in spectroscopy that we offer and some of the applications and challenges that they have. But also I want to talk a little bit about where our own spectrophotometers are used to help develop and improve the optics and photonics products that are used both in our products and also other products in the life science sector. And so that's covered in these orange boxes. I'll talk a little bit about external end users and operators of our life science instrumentation, and then talk a bit about the internal R&D and production challenges and interests that we have in the photonics and optics area to improve our life science offering and instrumentation. I'll be talking mainly about, obviously, spectroscopy. So that's UV-Vis, UV-Vis NIR. Infrared, we have fluorescence products and Raman products. I'll be touching on those. But Agilent was also quite deeply involved in offering solutions to the applied markets, life science markets, clinical and diagnostics testing. The division that I represent here in Melbourne, Australia, is the birthplace of atomic absorption spectroscopy, and it has grown to include molecular spectroscopy. So down here in Melbourne, we look after the atomic spectroscopy and the molecular spectroscopy. The first application in life science offerings that I'd like to talk about is a very, very common application across life sciences, predominantly in the pharmaceutical markets, is UV-Vis spectroscopy. It's really a workhorse within a lot of laboratories across the world in the pharmaceutical labs and the research labs, for doing very simple identification of pharmaceuticals and active pharmaceutical ingredients. We perform both the qualification of those, identifying differences between them, and also quantifying APIs as well. Our UV-Vis instruments are also used for protein stability, RNA, DNA ratio determinations, and the denaturation of nucleic acids as well. A lot of the important attributes of a spectrophotometer in this sense is temperature control. Obviously, a lot of the protein studies, which are temperature dependent, require studies to be performed at living temperatures around 37 degrees Celsius and higher. And so including temperature control in a spectrophotometric measurement is a critical attribute of instruments. A lot of the industry that we sell into in life sciences have a fairly strong focus on throughput for these kinds of measurements, so efficiencies in getting a large number of samples through, as well as photometric certainty in the results, so very high photometric performance, which would include things that have been touched on or spoken about by previous speakers, such as good resolution, high dynamic range, and stability and good wavelength accuracy. It's a little out of the scope, I think, of maybe this group here, but there's a strong interest for our life science instruments and offerings to have the appropriate electronic records, something that is important for data management, for electronic signatures, and for control of method development. The spectrophotometer that is pictured here is a Kerry 3500 spectrophotometer. A lot of the samples that go into a UV-Vis instrument go into cuvettes. And part of the novelty in the 3500 platform is to be able to measure 8 channels or 8 cuvettes simultaneously. And that's quite a breakthrough that was achieved by modularizing the design and splitting the engine and the sampling area. And that was done with some advanced photonics offerings like waveguides and 8-channel splitters there to achieve that. And that also allowed us to independently change the temperature of pairs of cell at 4 different temperature locations across the eight cells. We also heard just a moment ago as well about the importance of being able to bring the measurement to the sample as opposed to bringing the sample into the laboratory. And there was some really interesting data from Julia there in the milk industry and some of the important things here. I'd like to just, I guess, show you some of the novelty that our end users have found very critical, aligned with the idea of bringing the measurement actually to the sample. Two products here. The infrared spectrophotometer at the bottom, which is a handheld portable device that allows you to bring basic FTIR to a sample in the field. And also a Raman spectrophotometer, which is handheld, as you can see in the pictures on the right-hand side here. And it uses a novel technique in Raman spectroscopy called spatially offset Raman spectroscopy. And that allows the Raman signal to penetrate and get through barriers such as paper, such as polymers, such as glass, and to be able to measure the active ingredient or the product behind that barrier in the vessel that arrives in. That obviously has huge benefits in being able to measure at point of a receipt of the product in the loading docks in the incoming goods area. And it also allows for the prevention of contamination of products. It speeds up analysis by not having to move product into laboratory as well. And from an optical perspective, the miniaturization of instrumentation in this life science area is quite important. So obviously smaller detectors, high sensitivity, and equally important is the sampling design for those kind of applications. Just moving on now to more of the photonics in the life sciences. Our highest performance UV-Vis and UV-Vis NIR spectrophotometers are sold into the optical industry, where those optics eventually are used in a lot of the life science instrumentation. Two very clear examples, and I think it's been touched on in the earlier talks, are both Raman and fluorescence, where the excitation wavelength, typically a laser, must be suppressed in the measurement signal. So for Raman, you want to look at suppressing the main laser line frequency and just look at the Raman signal, and that's very close in terms of wavelength or wave number from that excitation. So having very strong blocking optical filters with high optical density or high absorption, but also very high transmission, very close to that, requires filters of exceptional performance. And that's something where our UV-Vis and UV-Vis NIR spectrometers are used. Some other trends, and I think I've heard this earlier as well, both in the drive to miniaturization and the mobility of instruments, is for smaller optics and for optics that are of unusual morphology or shape. That means beam splitters, prisms, gratings, and some of the novelty in the spectroscopy has required us to move detectors around samples so that we can detect light being transmitted or reflected at multiple angles. There's also a huge drive as well for upscaling and large operational efficiencies. So robotics that are needed to measure multiple samples, or for instance, measure multiple points on large wafers, optical wafers or semiconductor wafers that are up to 200 millimeters in diameter. If we look at internally now within Agilent and our R&D teams in terms of the kind of optical or photonics products which we have historically seen benefit the solutions that we provide to our end users and advance the solutions that we can provide to the life sciences area. It really covers those four aspects in the blue box. It's the source, obviously the key component of a spectrophotometer, the method of detection, the light delivery system, and also sample preparation. So from a source perspective, sources have changed in spectroscopy over the last few years. There's been large advances in the implementation of xenon sources that are extremely high intensity and have extremely rapid flash rates. There's also quantum cascade lasers, which are being employed in our imaging systems. For instance, the laser direct infrared imaging system uses a quantum cascade laser. So just two basic examples of improvements in sources recently. We also see from a detection side some challenges on the scarcity of rare earth elements and also some regulatory pressure, for example, on lead sulfide or lead. Both in electronics with RoHS, for example, back in 2016, but that also has some impact on the detectors that we can offer as well. Light delivery is an area that's moving quite quickly, and by that I mean obviously the optics that deliver light to the sample and to the detector. Freeform optics, fiber optics or waveguides are certainly being used more predominantly in a number of applications, as well as plastic optics. Sample preparation I won't touch on in a great deal, but obviously that's a significant part of the overall measurement systems for spectroscopy. I'll leave it just there. I really just wanted to touch at a very high level on a number of instruments and the optics and photonics solutions that back those for Agilent. Happy to have any questions.
Thank you so much, Travis, and double thank you because I know it's really, really late for you, so I really appreciate that. We all appreciate it, yeah. So actually, we have a question from Arnaud from Lyolix International. So Arnaud, could you please tell us your question and also tell us a little bit what Lyolix International can do in this for the spectroscopy?
Yeah, hello, good night, I would probably say to you, Travis. I had a question because you had the great results on this sensor measurement system that you brought into the field. So you listed a lot of requirements, a lot of capabilities and possibilities there. But what was the one really killer feature that really got you the customer to really get into this system and to really buy it and use it? If you can name one.
Sorry, the killer
feature.
application.
Yeah, the killer feature for the system you brought into the field.
Yeah. I think the killer feature from a technology perspective is the spatially offset Raman, which is the enabling technology to be able to measure through the containers, and then the value that brings those customers is to be able to measure incoming goods in the receipt area of a pharmaceutical company without having to open up packaging. So there's a safety aspect where the operators don't need to come in contact with the material. There's a cost aspect because the samples could be measured very quickly and rapidly without having to be sent off to the lab, measured in the lab, and the delay in getting the results back. So it's essentially a safety and a financial benefit to the end users.
Yeah, so sounds like a great feature. A really good system. Thank you.
You're welcome.
Yeah. So Arnaud, tell us what Lyolix can do.
Yes, if I can share the screen. If there are no other questions for Travis, yes.
Yeah.
Otherwise, we can still go afterwards. Yes.
So here we go. Here we go.
Yes. Should
3,
go
2,
to
1. Presentation mode.
full screen.
There.
Yes, welcome everyone. My name is Arnold Everhard. I'm for Linux International. Linux International, we have our own, our silicon nitride. We invented Triplex, our silicon nitride platform for integrated photonics. At Linux International, what we do, we can do all the way from design to the manufacturing, assembly and packaging, all the way the modules for customers, OEM customers and other components, or other. So we're not an end user, we build systems and modules for our customers. Here, of course, we also have a lot of experience in spectroscopy. We've done a lot of things in the past. On the top left, you can see one of our beautiful chips. It's a beautiful photonic integrated circuit. White light coming in and split into all the different color bands. It's a beautiful picture, a beautiful application. And what I want to talk about here in this meeting is also about our EU project Forever, where we're actually building a flow cytometer system. Well, a flow cytometer system is really you have a microfluidic channel, particles coming in, focusing, and then you have the light integration, and then you have all kind of scattering and fluorescence from all these particles. Of course, this is typically a bulky system. Here we have a laser, free space optics. We can bring all of this together into a photonic integrated circuit. And for this, we actually build a chip here, and we have the light coming in all the way. And we can actually have a small little port with which you can zoom in, bring the top right and top left together into one little close and beautiful image, where we can actually not only just have photonic integrated circuits, not just light on the chip, but also do more of it. We can integrate it and build all kind of custom components onto and into the photonic integrated circuit. So this is actually courtesy of our partner, Fixed Photonic Assembly. They made a beautiful photograph and printed some 3D lenses on the circuit, where you can really have the light going from the circuit towards a capillary microfluidic system and being detected at all the different angles, and then with all kind of on-chip light filtering, create a beautiful flow cytometer in a miniaturized system. The system is just being made. We're still doing the test. So and of course afterwards needs to be a product. And also asking here around, because in this consortium we don't have an end user yet. So of course this is technology showcase. And of course, if there's more interested people to bring this even further into the market, we're always open for discussions.
Thank you so much. And this is one of the things. When is going to be the peaks going to be a replacement into the bigger systems? And talking about the other thing is like, okay, there is a lot of portables, and that's what we are moving towards now. So a little bit into the portables, but also the need of having miniaturization. It could be miniaturization in the surgery room, but it could be miniaturization in the field, and who actually can talk about the spectroscopic modulus and optics that defines the system performance in just a block is Bark Rose from Ipsen Photonica. So, Jark, the floor and the attention is all yours.
Thank you, Helena, and thank you for inviting me for this presentation. So let me see. So I'm from Ipsen Photonics. We do gratings and spectrometers for industry. Let me say up front that we have a more classical approach with bulk optics. We do our own grading and put spectrometers together. And I'll speak about miniaturizations and some of the pitfalls and design guides, just real overall.
Here you are.
Yeah, I think it's coming now. Sorry for that.
Yeah. Yes.
So spectroscopy for life science, you typically have a light source, a sample, and then you measure with a spectrometer. This is a transmission. It could as well be a reflection geometry. So the spectrometers are used as spectral detectors in the instruments, and in life science, it's typical, and we've heard that before many times today, to measure absorption, fluorescence, Raman, optical coherence tomography. Those are all some of the common technologies applied in life science. And why miniaturization? It enables portable or even handheld or wearable devices. To some just wants more space efficient, but some want to go like Natalia wants to do something really compact, and I'll try and come back to that. So in a classical spectrometer, how do you miniaturize? So the size of the spectrometer scales with the detector length and the grading period. Those are kind of laws of nature. So to miniaturize, you want to use a short detector and a high groove density grading. And actually, for us to miniaturize, the availability of small detector housings has actually been a limiting factor for many years. So you could get small detectors, but they all came in very big standard housings. But nowadays, you can get them much more compact. And then you need high throughput, so you choose a high efficiency transmission optics with lenses, transmission gratings, because that will allow you to have a higher numerical aperture to counteract the reduction in sensitivity due to miniaturization. So I think I posted a LinkedIn message following when you made an advertisement for that, because everybody wants it smaller, but nobody wants to pay for the penalties.
Yes.
And it definitely have consequences, and that needs to be balanced also when you start to think of, do you want to go with bulk optics, do you want to with filter technologies, do you want to do with PICs? There are a lot of things to consider. In a classical spectrometer, the easy back of the envelope is that the throughput scales with the area of the grading. So that's just squeeze it down, half size grading, half throughput, just a rule of thumb. So some of the consequences are low sensitivity throughput. And we heard before Agilent, they were saying we need throughput to speed up measurements. So that naturally leads to low signal to noise ratio. Often we've tested a lot of these compact devices. There's a really high level of stray light, but that may not be an issue depending on the application. Some of them are very sensitive to light coupling. Some have limited configurability. You take one config and you buy 10,000, so it has a fixed configuration. Some have very large thermal spectral shifts, and some are very sensitive to vibration and shocks. And again, we've taken the approach that we're good at. And one example comes here is our Pebble platform. Fairly compact, as you can see on the right-hand side, comes in Vis near. And we also have a near-infrared version. We still stay with the scientific grade, high-quality detectors, Hamamatsu detectors, and we do the same with the near-infrared. So you may ask, I think you can see it here. That's I'll put it closer to the camera. That's the Pebble that I just showed you. But we can also do smaller. And actually this, what I have here next to it, maybe you can't see it from my bulky fingers, but that's a Raman spectrometer, 800 to 1100, still made with bulk optics, the size of the SMA. This leads to a detector with only a few millimeters in length. So signal to noise is really, really an issue. But again, for us to miniaturize, it's not really a big thing. Somebody just needs to tell us what detector to use that would work with that compact device. So yeah, that concludes my presentation. Thank you for your attention.
Thank you so much. So yeah, so it is, I mean, going back to the detectors, is going to be also lack of materials, so new detectors, and also adoption of different detectors that we typically don't use for some different things. But we are continuing moving into the portable systems. Okay, so we have in the room Michela Scienza from Pioneers, and they focus on portables near-infrared systems where constraints in power, size, and environment are critical. So, Michele, the floor and attention is all yours.
Hey, Lena. Hi, everyone, and thank you for inviting us and me to talk at this meeting. I'm excited to be here and to hear those nice talks. And I'll start with sharing the screen first of all. and a bit of introductions, because we are changing a bit the paradigm of how we are doing measurements and what we are interested in. We actually don't use directly any spectrometer in the device that we develop, but we do kind of limited spectroscopy using different laser sources. So we dig now into tissue hemodynamics and tissue optics. So we look at highly scattering media, so our brain, our muscles, and eventually also we have some different applications in fruit and vegetables monitoring. And so anything which has big scattering properties, it's suitable for those kind of measurements. So focusing on tissue optics, the penetration depth of the signal that we deliver to the tissue reaches approximately 2/3 centimeters. And we are not interested in making images of what we measure, but we are interested in retrieving quantitative values of concentration of oxy and deoxyhemoglobin. So sticking to the hemodynamics properties and of our tissue, those are extremely important when we look at the brain, especially because so cerebral hemodynamic impairment are within the top 5 leading cause of deaths, and there is no actual daily use technique in the clinics to monitor those kind of variation other than invasive procedures. Instead, on to the muscle hemodynamics, muscle hemodynamics impairment is often related to chronic pain and also many other symptoms related to very harsh pathologies in general. So it's good to be in this conversation because actually when we tackle the clinic, talking about NIRS, so near infrared spectroscopy applied to clinical usage, there is a quite broad malcontent, so quite broad difficulties in the clinical scenario with actual clinical devices based on continuous wave NIRS. So NIRS using LEDs and standard photodetectors to retrieve the variation in concentration of oxy and deoxyhemoglobin. There is actually very an actual problem on accuracy, consistency of measurements, and reproducibility across same device measurements and different devices out there. So current medical devices have big limitation in retrieving reliable results. So we tried to tackle this issue, introducing a new technique and technology to this field. So we are talking about time domain NIRS. And the domain of time here goes down to the picosecond level. So where we use pulsed laser sources of average power of around tens, let's say 5 up to 10 millivolts, and single photon detector. So after delivering the light to the tissue, we collect the photons in 2 centimeters apart or 2/3 centimeters apart. And by looking at the shape of the temporal modification of the shape of those lasers after propagating into the tissue, we are able to retrieve the concentration of oxygenated hemoglobin, deoxygenated hemoglobin, so tissue saturation and total hemoglobin, an index of the volume of the blood present in those microvessels. So we are mixing both the arterial blood and the venous blood present in the volume that we are measuring. And this is the NIRSbox device. So it's the device that Pioneers is commercializing for research applications. And as you see, we have the 2 laser sources, the single photon detector. Everything is delivered to the tissue throughout optical fibers and optical probes that can deflect light 90 degrees to reach the skin surface. And the time-correlated single photon counting board that we developed in-house must have down to 10 picosecond resolution to retrieve a good temporal information for the laser sources after propagation. So why do we work in the domain of time? Because photons arrival time encodes penetration depth. So that's why we're able to reach those depth inside the tissue, so up to 2.5/3 centimeters. Late photons in the curve will bring us information about about deeper layer inside the brain. For example, here the brain cortex. While early photons will bring us information about shallow layer or extracerebral information. And that's important because when you are looking at functional activation, for example, here we see that when the subject sit down, we have a response in the surface, which is the bottom right, that encodes an increase of total hemoglobin due to the increased pressure due to the orthostatic stress, while on the top left, we see what's called actual functional brain activation. So related to the neurovascular coupling, oxygenated hemoglobin going up and deoxygenated hemoglobin going down. And we are able in the same spot to disentangle those two information and have a clear picture of what's happening in the brain. So that's how the actual data looks like in clinical environment where long-term monitoring is needed. So we have quantitative information of oxy, deoxy, hemoglobin, total hemoglobin, and tissue saturation. Moving on into some of the clinical application, here is a recent study that was performed in Italy on more than 350 pediatric patients from 0 to 18 years old, where the aim here, since the technology is very new and it is within the first able to record quantitative information about deep hemodynamics, we managed to retrieve the percentiles values for the pediatric population. This was on healthy subjects. This will enable the translation to pathological subject, and some papers are already out. One of them is specifically on a lower respiratory tract infection and the effect on prefrontal cortex hemodynamics. And here instead we see a environment shift when we look at the pediatric intensive care unit or the neonatology intensive care unit, neonatal intensive care unit, and a comparison with the current NIRS system. So CW NIRS is the current medical device standard, and TD NIRS is our technology implemented in Pioneer's devices. And here we see that before and after transfusion, so those little babies were underwent transfusion, blood transfusion, because they could not oxygenate well their own blood. And what happened here is that with the state-of-the-art research device that we have, it's possible to actually define a threshold and see if the transfusion was actually usable or not in those kind of patients. So different applications are instead into the sports environment and the health of our muscles and of our system related to the oxygen consumption inside big muscles or even smaller ones. So here is an example of measurements on the vastus lateralis during cardiopulmonary test. And what can be done here is to look at the hemodynamics of the muscle during the exercise. Here is a difference of, let's focus only on the yellow and the green curve related to the total hemoglobin content and saturation, tissue saturation of the muscle that's making the effort. In the warm-up time, we see tissue saturation oscillating between 60%, while in the exercise, we see the average tissue saturation dropping and not a clear correlation with the actual movement. While we see the hemodynamic variation shifted to the total hemoglobin, so the volume of the blood coming into the small vessel inside the tissue. While the blue one, it's a kind of an index of the motion that's happening. This is the average over 3 different pedaling cycles. And these can be translated to pulsatility, for example, of both brain pulsatility and muscle. Why quantitative values matters, and this is what time domain NIRS can actually bring into the new paradigm of NIRS, is that they can tell us a lot about the tissue, both structure and potential. Here it's a difference. Here is a study made on the difference between young and elderly subjects, and we see that total hemoglobin, so the hemoglobin volume in the muscle, is much higher in young subject compared to elderly one. And these happen as well as the vasoreactivity, so the speed at which the muscle reacts to external environment. Here, the example was a vascular occlusion test, where we see a much faster decay in young subjects rather than adult subjects. So these can be highly related to aging and how well we are aging in our own lives. I'm going to wrap up with a glimpse of where Pioneers devices are around the world and to which laboratories we are collaborating, and a glimpse to the future. So we are now working into the miniaturization of those systems, and this is where we are looking for new users and partners into this task and challenge, and trying to make those devices that now are as big as a book, let's say, into some truly wearable devices. And thank you for your attention, and I'm here to address any questions.
Thank you, thank you, thank you so much, Michele. I mean, it's like a really big, impressive change since you started. So it was only the oxygen detections for the very beginning,
Yeah.
and this is no, it's really impressive. So what do you need to get into the right miniaturization, and what is going to be your limiting signal to noise? Because this is what's part of what is going to be part of the miniaturization on the size.
Yeah, so one of the limiting part right now is related to so other than what we've already been talking about. So money is one of the first limiting factor, of course, but so clinical adoption is one of the one of the walls we have to hit when we come out with new devices. So having partners in the clinics who actually are willing to test new systems, new devices, and see if they can actually be more useful for their clinical routine. It's something that we are looking for every day. So new users also, we are happy to give out demos or collaboration related to some specific projects, while switching into the miniaturization. We actually developed our detector and laser chain. So having an idea on how we could integrate it in all together, this could be very useful and valuable if we find any partners in this direction.
Okay, so it's basically more for the integration. So if I ask you, give me your Santa, because we are close to December. Never, but give me your Santa list. Give me your 3 things that you want.
So let's say the first one would be clinical collaborators. The second one would be having someone that can collaborate with us to co-develop some integrated circuitry for some wearable solution. And the third one would be to, well, that's already enough. I mean, the second one can be split in 2 parts, one related to lasers and one related to detectors.
Yes. Okay. Yes. Absolutely. So you know that there are a lot of people here in the room that can help you with that. So yeah, and I would be happy to connect you. And after we finish here, we can stay a little bit for talking if you want. But I would like to move to the very last speaker of today, and it's like, what happened when I know what I want to do, but I need to do my prototype. So something who can help with that is Darius from Clear Photon. So Darius, the floor attention is all yours.
Thank you very much, Helena. So what clear photon does is what Helena already mentioned, this station for prototyping. And can you see my screen?
Yes.
Perfect. Yes. So I selected this one study here by Mackenzie et al., who developed this microfluidic chip, and which was meant for SIRS. So they manufactured the glass with femtosecond lasers that they had in their lab. They did this amazing silver nanostructure where they used femtosecond laser for doing that. To perform some measurements for this Raman spectroscopy. And then not everyone is as lucky as they are to have this expertise to build the entire system and to have like femtosecond lasers in-house. So Clear Photon is now like coming forward to help also other guys, other labs who would like to do, for example, something similar. So Clear Photon is this desktop micromachining station that can do prototyping. So you can think of it as like a 3D printing, but subtractive, not additive. And then like either you want to do this like microfluidic channels, cut glass, or do some precise drilling. You can, match it with like any laser on the market, do it fast and cost effective, of course. And most importantly, it's like safe, so you don't have to have expertise on the lasers, on the integration. You just, buy it and use it.
Thank
you very much, Darius, and congratulations. Congratulations on what you are achieving with Clear Photon. It does look like you have something fantastic. You said that you can actually put any laser in the system. Is there any particular partner that you're looking for that you would like to be connected after this meeting?
Yes, so definitely. So this device is an open platform for any lasers. So it's not only about micromachining, but it can also be about marking, for example. So any cost-effective laser source that's on the market might be like a very good match for this cost-effective laser station that Clear Photon is offering. So
Darius, a couple of weeks ago, we had a great meeting at Corning, New York, hosted by Xavier Lafosse, and there we talk a lot about the use of femtosecond lasers for the next generation glass processing. I'm pretty sure this setup is actually very interesting to some of the R&D partners. I would like to thank you, but also thank everyone who has contributed to the last 1 hour and 52 minutes of discussion. The most important part of this meeting was to make connections. So all of you prepare to be introduced to many people by email. All we ask you is that you follow up on those introductions. From the bottom of my heart, I would like to thank Dr. Helena Diaz for a fantastic performance, and all of you for choosing Optica as your platform for business. Okay, so thank you very much. I would like to first ask my IT department to cut the YouTube stream. So please disconnect it. And now this becomes a private meeting for all of us having a private